Hello, I'm Jacob Evarts, and this is my poster on propagating putative prion states in RNA-modifying enzymes in budding yeast. Prions are misfolded proteins that can cause other proteins of the same type to also misfold. Protein shape is closely related to a protein's function. So by changing the shape of the protein, you also often change the function of the protein by revealing active or binding sites that were previously hidden inside of the protein. Additionally, once there are enough prions present in a cell, they begin to agglomerate or aggregate. Previously, prions were connected with disease and detrimental conditions such as mad cow disease and Creutzfeldt-Jakob disease. The Garcia lab and others have been challenging this notion by showing that prions can also act as an important form of epigenetic inheritance, such as with the big plus prion that was recently discovered in yeast that allows cells to grow faster and larger than the normal or naive counterparts. Previous work has shown that protein overexpression and exposure to environmental stress can cause naive proteins to flip to prion states. The goal of my project is to use these methods to conduct and analyze high-throughput screens testing many different RNA-modifying enzymes crossed with many different stress conditions. RNA-modifying enzymes were chosen because they are a highly conserved class of protein and are related to many different human diseases, including cancer and neurological disorders. Due to the large amount of data produced by high-throughput experiments, I've been using machine learning techniques to process the data, such as neural networks and regression trees, and hope to eventually predict putative prion states using these methods. The results have been showing so far that prions appear to be a common form of inheritance in yeast. At least 9 of the strains tested have shown significantly better growth when they had a protein previously overexpressed than those that did not. These putative prion states suggest that prions could be a widespread mechanism of inheritance and not just detrimental diseases. We hope soon to have fluorescent images of these proteins to observe whether they exhibit a similar aggregation pattern as other known prions. Additionally, we hope to test the human orthologs to many of these proteins in similar yeast assays. I would like to thank the VPRI Fellowship for making this research possible, and thank you for listening.